monocytogenes, is a gram-positive bacteria that cause listeriosis. Listeria was discovered by a Scottish doctor, J.H.H. Pyrie, who named it in honor of a British surgeon named Joseph Lister. It's also called monocytogenes because when it was inoculated in rabbits, their monocyte levels increased. Alright, now L. monocytogenes is a rod-shaped bacteria with a thick peptidoglycan cell wall. So when gram-stained, it takes in purple dye, making it a gram-positive bacteria. It's catalase positive and oxidase negative, which means it produces the enzyme catalase, but not oxidase. L. monocytogenes is a facultative intracellular pathogen, meaning it can live both outside or inside of its host cells. It doesn't form spores, and it's facultatively anaerobic, meaning it can survive in both aerobic and anaerobic environments. Now, when L. monocytogenes is cultivated on blood agar medium, its colonies cause beta hemolysis, also called complete hemolysis. That's because it produces toxins called beta hemolysins, which hydrolyze the hemoglobin within red blood cells to transparent yellow-colored byproducts. L. monocytogenes is a motile bacteria with a very interesting way to move that depends on both its location and the temperature. In an extracellular environment, this bacteria moves by beating its flagella, creating a characteristic tumbling motility. But this is only possible at 37 degrees Celsius and below. That's because FLAA, the gene that codes for flagellin, which is the structural protein that makes up the flagella, becomes downregulated as the temperature rises up to 37 degrees Celsius. So above 37 degrees, there's reduced production of flagellin proteins, meaning no flagella being made, rendering L. monocytogenes non-motile. In an intracellular environment, L. monocytogenes moves by an actin-based motility. It starts with this bacteria producing a protein called actin assembly-inducing protein, or ACTA, which recruits small actin filaments at one end. As more and more actin filaments get recruited and polymerized behind the bacteria's end pole, the bacteria propels forward, like a rocket. L. monocytogenes is a foodborne bacteria, meaning people get infected when they eat contaminated food, especially unpasteurized dairy products and cold deli meats. And this bacteria is known to survive even at very low temperatures in the fridge. Once this bacteria gets into the gut, it uses its attachment proteins called internalins to attach to the host's receptor proteins, like E-cadherin located on goblet cells of the intestinal mucosa. After attaching to the host cells, this bacteria is slowly engulfed by the cell membrane, which invaginates to form a sac on its inner side. The sac then separates from the actual cell membrane, forming what's referred to as an internalization vacuole. Inside the vacuole, Listeria releases listeriolysin O and bacterial phospholipases, which are enzymes that degrade the vacuolar membrane. This releases the bacteria in the host cell cytoplasm, where it starts using the host cell resources and food, and multiply into many identical bacterial cells by the process of binary fission, which means every bacterial cell splits into two identical copies. On a side note, if this sounds similar to mitosis, well, it is. But the term binary fission is used to describe division of prokaryotic cells, which don't have a nucleus, and therefore some steps in replication are different from mitosis. Inside the cell, L. monocytogenes moves around using the actin-propelled motility, and when it reaches the cell's membrane, it stretches it towards an adjacent cell. When it reaches the adjacent cell membrane, that one invaginates as well. So Listeria ends up wrapped in a double-layered sac. Then this sac pinches off inside the adjacent cell, as a double membrane vacuole. Inside the vacuole, Listeria releases listeriolysin O and phospholipases to break free into the cytoplasm, repeating the invasion process over and over to invade more adjacent cells and eventually get into the bloodstream. From the bloodstream, the bacteria spreads to other organs, resulting in disseminated listeriosis. This is most commonly seen in people with weaker immune systems, like neonates, the elderly, pregnant individuals, and adults with immunocompromising conditions, like diabetes, malignancy, or an HIV infection. Under these circumstances, Listeria can affect the liver, resulting in a liver abscess, or move up to the brain through the blood-brain barrier to cause meningitis, which commonly occurs in neonates, the elderly, or adults with diabetes. The worst case scenario in pregnant individuals is if the bacteria crosses the placenta and gets to the fetus. This may result in a spontaneous abortion or stillbirth. If the fetus survives to term, it might be born with neonatal listeriosis, which can present with neonatal sepsis and meningitis. Now, when listeriosis is limited to the intestinal mucosa, it presents with signs of gastroenteritis like low-grade fever, diarrhea, and vomiting. With disseminated listeriosis, there's a high fever, muscle pain, and additional symptoms depending on what organs are affected. With liver abscesses, there might be abdominal pain and jaundice. And with meningitis, symptoms include headache, stiff neck, and altered mental status. Neonatal listeriosis classically presents with fever, respiratory distress, and a specific skin rash consisting of broadly spread, small, pale nodules referred to as granulomatosis infantiseptica. Diagnosis is done with cultures from a biological sample, which varies depending on symptoms. So a stool culture is done for gastroenteritis, a blood culture when bacteremia is suspected, cerebrospinal fluid cultures for meningitis, and amniotic fluid cultures are done to diagnose a transplacental infection. Imaging studies like an ultrasound or a CT scan can be useful to identify liver abscesses. Now, when listeriosis is limited to the gastrointestinal tract, treatment usually comprises of supportive measures like hydration and rest. Disseminated listeriosis, however, is treated with intravenous ampicillin combined with gentamicin. In people with contraindications to these antibiotics, specifically pregnant individuals, or those who are allergic to ampicillin, meropenem is a good alternative. Alright, as a quick recap. Listeria monocytogenes is a gram-positive, rod-shaped, facultative anaerobic and beta-hemolytic bacteria. It's also a catalase-positive and oxidase-negative, facultatively intracellular pathogen. Listeria has a tumbling motility in the extracellular environment, and actin-based motility intracellularly. Most commonly, it causes listeriosis in neonates, immunocompromised adults, and the elderly. Listeriosis can present with low-grade fever, diarrhea, and vomiting, which can worsen to high-grade fever, jaundice, and altered mental status in the case of disseminated listeriosis. Listeriosis is diagnosed using cultures, and imaging studies like a CT scan might help identify liver abscesses. Treatment is done with a combination of ampicillin and gentamicin, or just meropenem in pregnant individuals or those who are allergic to ampicillin.